Assalamu alaikum wa rahmatullahi wa barakatuh. This is video number 14. It is the fourth set of videos. Each set of videos correspond to one lecture. In this video we talk about the second type of lipids, or the second class of lipids, which is the class of non-saponifiable lipids. This class contains steroids and terpenes. This class is characterized by the absence of fatty acids. No fatty acids occur in these lipids, therefore they are not saponifiable because saponification depends on the presence of fatty acids. Steroids are actually diverse. Chemically they are diverse, however they have in common these four cycles. These four cycles in chemistry, they are named perhydrocyclopentaenophenanthrin. You are not supposed to memorize this name. Just remember that the same cycles are shared by all steroids. Here we have cholesterol, we have testosterone, we have estradiol. All of them share 3 cyclohexanes, 1, 2, 3 cyclohexanes, and 1 cyclopentane. The difference among the diverse steroids lies in the radicals, in the chemical groups that are linked at the diverse or the different positions. Here, for instance, we have methyl, but here we have this long hydrocarbon chain, while here we have only this hydroxyl group. Again, I repeat, you are not supposed to memorize the chemical structures. Just remember that all steroids share 3 cyclohexanes and 1 cyclopentane organized in this manner. However, you must be able to give some examples, and the most important examples of steroids are cholesterol and steroid hormones. Testosterone is well known, estradiol also. Notice the difference between testosterone and estradiol, two hormones that have totally different functions. The difference between them is just in this chemical group, methyl group present here, absent there, and this hydroxyl group is replaced by the carbonyl group. Only these two chemical differences make a huge difference in terms of activity. Vitamin D is derived of cholesterol. It is an example of steroids also. Cholesterol, you notice that cholesterol is a molecule that has all of this region, the region I'm indicating now, all of this region is made up of carbon and hydrogen, therefore it is non-polar. The only polar radical is the hydroxyl group here. We schematize cholesterol in this way with a long, large hydrocarbon part and a small hydroxyl group. This makes cholesterol somehow amphipathic, slightly amphipathic. And because it is slightly amphipathic, it fits in the membrane. Where is the membrane? From this line to this line, this is the membrane. The membrane is made up of 2 layers of phospholipid. This is the first layer and this is the second layer. I know that these tails are known for you. In the previous videos we explained that phospholipids and glycolipids have hydrocarbon tails that are non-polar, and they have hydrophilic heads that are represented as circles. Here cholesterol fits in one layer. All of the regions that are hydrophobic assemble together. The hydrophobic tails assemble together, and in the other layer the hydrophobic tails assemble together. So cholesterol is a component of the membrane, not in all living things, especially in animals. It is especially present in animals. It may represent up to 50% in molarity of the membranes in a lipid, of the lipids in the membrane of an animal cell. We know that cholesterol is abundant in eggs, in meat, especially in the liver, in cheese, etc. It is known that excess of cholesterol is harmful to the cardiovascular system, to the circulatory system. However, a diet with 0% cholesterol is not healthy at all, because cholesterol is indispensable for the production of vitamin D. Cholesterol is a precursor of vitamin D. Cholesterol is a precursor of bile salts. Cholesterol is a precursor of the adrenal hormones, the hormones that are secreted by the adrenal glands on the top of the kidneys. And of course, the cholesterol is important for the production, for the synthesis, for the synthesis of sex hormone, testosterone, estradiol. So our diet must be balanced, must contain a certain amount of cholesterol. The excess of cholesterol is harmful, but we must have a balanced supply of cholesterol. Because cholesterol is present in the membrane, it will affect its fluidity. We don't emphasize much on the effect of cholesterol on fluidity. Just remember that it is a regulator of membrane fluidity. This membrane is fluid. Fluidity is important. It is an important feature of the membrane. We will consider membrane fluidity later on in chapter 4, inshallah. Okay, now let's consider some other lipids that are not saponifiable. Terpenes. Terpenes are polymers of propene, CH2, CH, CH3. Vitamin A, for instance, is a terpene. Vitamin E, vitamin K, lycopene. Lycopene is the pigment that is responsible for the red color of tomato fruit. Vitamin A is derived of carotene. It belongs actually to the family of carotenoids. The family of carotenoids is a family of pigments that are responsible for diverse colors of carrot and other plant organs. Because they were discovered the first time in carrot, they were named carotenoids. So it is a range of pigments having different colors. All of these are carotenoids. We notice that overall a terpene may be hydrophobic, with a very small hydrophilic chemical group. Sometimes we have a pigment that is totally hydrophobic, lycopene, the red pigment responsible for the red color of the tomato. It is totally hydrophobic. This is why when we have to use tomato paste, for instance, or tomato juice during preparation of food, we notice that the oil droplets they get colored in red. The red color of the oil droplets is because the lycopene pigments are hydrophobic and they are more soluble in oil. If I made a mistake, I correct it. Lycopene is hydrophobic. It is not hydrophilic. There are many other terpenes actually. You can be satisfied with these examples of terpenes, vitamin A, vitamin E, vitamin K, carotenoids, without memorizing their chemical structures, even without memorizing the propene structure. Just memorize some examples and the fact that they may be hydrophobic or hydrophobic with a certain small hydrophilic group. There are so many types of terpenes. We have limonene, for instance. Limonene, it is the terpene responsible for the specific smell and taste of lemon. We have, for instance, menthol, menthol بالنعناع. It is also in the family of terpenes. We have pinene بالصنوبر, etc. Another set. So terpenes are extremely diverse. We are not going to know about the details. We are satisfied with the examples I mentioned. The question I would like to answer now is about behavior of lipids in the cell. If you remember, at the beginning of chapter 2, I told you that water is the major component in all living things. And lipids, most of them are either nonpolar, totally nonpolar, or amphipathic. The question now is, how do lipids behave in the cell, which is an aqueous environment? It is extremely rich in water. Behavior of lipids in water is important to be understood because it will help us understanding the structure of membranes. Remember, membranes are mainly made up of phospholipids, glycolipids, sometimes cholesterol, etc. To understand the behavior of lipids in water experimentally, as well as inside the cell, we are going to take amphipathic molecules. They could be soap molecules, they could be fatty acids, they could be phospholipids, glycolipids. We take them, we add a large amount of water, and we perform vigorous shaking. Vigorous shaking will lead to a suspension because lipids are not soluble in water. It is a suspension or a heterogeneous solution. If we look at the structures that are formed in the heterogeneous solution, we notice that there are three types of structures that form when we perform mixing of the amphipathic lipid with water. The first type of structure, of course, you know that this is an amphipathic molecule with a hydrocarbon tail and a hydrophilic head, a soap molecule, for instance, a fatty acid molecule. This is the structure that simplifies the two tails of a glycolipid or phospholipid and the hydrophilic head. So we can schematize in different ways. What I represented as a vertical zigzag line in a previous video, it is now schematized as a straight line. It doesn't matter. We understand that there is a region that is hydrophobic and a head that is hydrophilic. So back to the result of mixing of amphipathic molecules with water, we get three types of structures. The first one is a micelle. A micelle is a microscopic sphere that has the hydrophilic heads at the surface of the sphere and the hydrophobic tails directed to the center. The hydrophobic tails are directed to the center. They create a zone that is devoid of water. They are joined with one another. I mean that the hydrophobic tails are joined with one another due to the hydrophobic interactions. We used the term hydrophobic interaction when we described the tertiary structure of proteins. Hydrophobic interaction is just an interaction between two chemical components or two parts of two chemical components that are both hydrophobic. They dislike water. They assemble together just because they dislike water. They don't want to have any contact with water. Everybody sees the micelles during the use of soap. When we use soap, water becomes turbid. Turbidity of water after using soap is because of the micelles. The micelles are the microscopic structures, the spherical and microscopic structures that correspond to this one. The greasy matter that may be present on our hands, the greasy matter is removed from our hands by being sequestered, imprisoned in the center of the micelle. This is a schematization of the 3D structure of a micelle. The second type is a lipid bilayer. When we mix amphipathic molecules with water, we get lipid bilayers. The lipid bilayers have the hydrophilic heads exposed to one side, the hydrophilic heads exposed to the other side, and the tails maintained with one another in a region that is devoid of water. Hydrophobic interaction stabilizes the tails with one another. This lipid bilayer has exactly the same structure as any membrane inside the cell. It can be experimentally formed when we mix amphipathic molecules with water. It is due to the chemical behavior of these amphipathic molecules. Now, if this lipid bilayer is large enough, it may fold back and form a liposome. A liposome is a kind of artificial vesicle. This artificial vesicle has a membrane. Where is the membrane? This distance represents the membrane, with the hydrophilic heads at the external surface, the hydrophilic heads at the internal surface, and the hydrophobic tails are assembled with one another. There are so many applications for liposomes in the field of pharmacy, in the field of medicine. It is not our topic. If you are interested, you can have an idea through the internet. In the next video, we talk about the last class of organic compounds, which are nucleic acids.